Hi, I'm Stephanie Zimmerman. I'm a scientist at Nanostring, and I'm going to tell you today about using Nanostring's Whole Transcriptome Atlas to study kidney disease in a spatial context. So kidneys are your body's filtration system, and when they're working properly, their job is to filter waste products from your blood into urine. And so the actual filtration site in the kidney is a structure called the glomerulus, which is a sphere of blood vessels and specialized cells that form a filtration barrier. And after the filtrate passes through the glomerulus, it goes into this very complicated network of tubules. First, the proximal tubules, which mostly absorb water and glucose and other small molecules. And then the distal tubules, which have more of a homeostatic role in regulating ion concentration. So when things aren't working properly, you end up with kidney disease, which is a very serious health problem. In the US, diabetic kidney disease is the leading cause of kidney failure. And this is because high blood glucose can damage the kidney, especially the glomerulus. One of the hallmarks of diabetic kidney disease is the loss of podocytes, which are a specialized cell type that helps form the filtration barrier. And so when the podocytes die, you end up losing proper filtration. Another hallmark of diabetic kidney disease is inflammation of both the glomeruli and tubules, as well as immune cell infiltration and fibrosis. So we were interested in studying the transcriptomes of diabetic kidneys in a spatial context. And the reason we were interested in doing these experiments spatially is because, as I mentioned, and as you can see in this immunofluorescent image, the kidney is a very highly structured organ, and these different structures express different genes. And so if we want to study the gene expression changes in these particular structures, you can't do that in a bulk transcriptome analysis. And so we really want to know how, these structures like the glomerulus, the proximal tubules, and the distal tubules are differentially affected by disease. Another reason to look spatially is that even within an individual, disease progression and pathology is very heterogeneous. So in a patient with severe kidney disease, most of the glomeruli will look very abnormal, but actually some of them will still look pretty normal. And we'd like to understand the transcriptional differences underlying this heterogeneity. And that's a very difficult question to tackle with bulk or single cell methods that lose the spatial and context and the pathology around the cells. So we used Nanostring's GeoMx DSP in order to profile the transcriptomes of selected regions in the kidney. And the way the DSP works is you hybridize barcoded RNA detection probes to your sample on a slide. And you can also add fluorescent antibodies that mark cell types or structures of interest to help guide the selection of the regions that you want to profile. So then you can scan the slide and you end up with a fluorescent image, and you can select your regions of interest on this fluorescent image. And then what the instrument does is that it illuminates just the regions of interest that you've selected with UV light, which cleaves the barcode off the hybridized ISH probes. And then the instrument collects those liberated barcodes into individual wells, one well for every ROI. And so then you can sequence the barcodes, and you end up with a count for every transcript for each ROI that you've selected. And then you can map those counts back to the image or to any annotations of that ROI that you have. So Nanostring recently launched the Human Whole Transcriptome Atlas. And this, the WTA contains probes for almost all protein coding genes in the human transcriptome, over 19,000 annotated transcripts. And so a few very high expression genes were intentionally dropped from the probe pool in order to reduce sequencing costs. And so the way these probes work, as I mentioned, they have a photocleavable linker that's attached to a barcode that identifies the ID of the RNA that the probe is hybridized to, as well as a UMI and Illumina adapters, so they can be sequenced on any Illumina instrument using standard workflows. So we used WTA to profile regions of interest in FFPE samples of human, normal, and diabetic kidney disease kidneys. And so this is an example of what 2 of the scans look like. And we also stained these slides with antibodies against WT1, which is a marker of podocytes, these specialized cells in the glomeruli. Pan-CK, which in the tubules marks distal tubules but not proximal tubules, and CD45, which marks immune cells. And so you can see all the ROIs that we selected overlaid over these images. If we zoom in on what the ROIs look like, one of the really nice things about the DSP is that it allows really flexible ROI selections. So you can just circle or draw a polygon around a region of interest. And that's what we did for the glomeruli. We selected individual glomeruli to profile. And as I mentioned, there's a lot of heterogeneity in disease pathology, even within a sample. So we actually had a pathologist annotate each glomeruli that we selected with the degree of pathology that it had. And we were able to choose glomeruli that were more severely diseased and those that were more normal. You can also select ROIs based on the expression of marker genes, and that's what we did for the tubules. So as I said, the distal tubules express pan-CK, and what the instrument can do is automatically segment this region into the pan-CK positive ROI and the pan-CK negative ROI. So you end up, you can separately profile the distal and the proximal tubules, even though they're quite close to each other in space. So overall, we profiled transcriptomes from 231 ROIs, and WTA also contains negative probes, which give you a measure of the background signal. So you can calculate the signal over background for each gene, and that's what I'm showing here on the right. I'm showing the SNR for every gene in rows and every ROI in columns, ordered by the highest expressed genes on the top. And so what you can see is that there's some genes that are expressed in every ROI, there's some genes that are only expressed in certain regions, and there's some genes that aren't expressed at all in any ROIs. Overall, we identified over 16,000 genes above background in at least 1% of ROIs, and over 7,000 genes above background in at least 20% of ROIs. So the first thing we wanted to look at with this data is to compare gene expression between these different structures of the kidney, and reassuringly, we can see that the ROIs cluster by the region that they came from, and also by disease status for the most part. We performed differential expression analysis of the gene expression in the glomerulus ROIs compared to the tubule, and the distal tubule compared to the proximal tubule. And I'm showing the results in these volcano plots on the right. And the colored genes in these volcano plots are known marker genes for specific cell types that were identified by single-cell RNA-seq. So you can see, for example, that podocyte marker genes in pink here are highly upregulated in the glomerulus compared to the tubules, as you would expect. And the proximal tubule marker genes are highly differentially expressed in the proximal tubules, and the distal tubule marker genes are highly differentially expressed in the distal tubules. And so that's great. We show that we can recapitulate, this known biology of the kidney. But we also identify many thousands of differentially expressed genes, some of which are novel. So next we focused specifically on the glomeruli, and we asked what changes during disease. And so what I'm showing here on the right is pathway enrichment for genes that are differentially expressed just in glomeruli between normal and disease samples. And so what you can see is that immune-related genes, such as complement, are highly upregulated in disease, which is really not surprising to us at all because, as I've said, immune infiltration and inflammation is a hallmark of diabetic kidney disease. And you can actually see that in these images here, where CD45 is marking immune cells. And you can just see there's a lot more CD45 in the diseased samples. For genes that are downregulated in disease, we saw a lot of kind of basic essential cellular processes enriched, such as translation, mRNA splicing, and mitochondrial respiration. And this suggests that the glomeruli are kind of losing their ability to perform these essential cellular processes and basic functioning with disease. One of the other interesting things you can do with WTA data is that you can couple it with known cell expression data from other sources, such as single-cell RNA-seq. And you can deconvolute the cell types and the fraction of each cell in each ROI. And so that's what I'm plotting here, the results of this cell type deconvolution, where each bar here is an individual glomerulus. And the colors stacked in each bar represent the proportion of each cell type in that glomerulus. And so on the top here, we have immune cells. And as I mentioned before, immune cells really increase with disease. And we can see that in the cell type deconvolution. The diseased glomeruli have higher immune cell fractions. One nice thing is that we can see which immune cells are most increasing in disease, and in this case it looks like it's mostly these mononuclear phagocytes in orange, these NK cells, natural killer cells in blue, and B cells in purple. And then below we have the actual cell types of the kidney themselves. And most of the cell types of the kidney are actually not changing all that much with disease, but I did want to highlight the podocytes, which, as I mentioned, a hallmark of diabetic kidney disease is the loss of podocytes. And you can see that here. The diabetic kidney glomeruli have about half the level on average of podocytes compared to the normal ones. However, what I hope you can see is that there is a lot of heterogeneity between individual glomeruli. So some glomeruli, such as kind of the ones in the middle, have no podocytes at all, whereas others, kind of some like the ones on the far right, have almost normal levels of podocytes. As I mentioned, we had a pathologist annotate all the glomeruli in our sample with the degree of pathology that they showed. And so if we now look based on this pathological annotation, we can see that the changes in cell type track with the degree of severity of disease. So for example, in a diseased sample, these mononuclear phagocytes increase in disease, but the glomeruli that are more pathologically abnormal have a much higher increase than the ones that are relatively healthy, even if they came from a diseased sample. And similarly, even in a normal sample without disease, the glomeruli that look abnormal have increased immune cell infiltration. And you can see the same for podocytes on the bottom left here. The podocytes get lost with disease, but this loss is much more severe in the more abnormal glomeruli. And so to illustrate this, I'm showing an image on the right of 3 glomeruli that came from the same diseased sample that are very close to each other in space, 2 of which were annotated as abnormal and 1 of which was annotated as healthy. And you can see that although these glomeruli are right next to each other, they actually are very different transcriptionally and have very different cell type profiles. So the abnormal ones have completely lost their podocytes and have more immune infiltration than the healthy looking one. So to summarize, I showed you today that the GeoMx Whole Transcriptome Atlas can profile the whole transcriptome of a single region of interest, any region of interest that the researcher wants to select. And we used this tool to study gene expression patterns in specific biological structures of healthy and diseased kidneys, and to look at spatial heterogeneity within a sample across diseased glomeruli. And I'd also like to add that this kidney data set is publicly available for researchers to explore, and you can download the gene expression data and the images from the Nanostring website. Finally, I'd like to thank the people at Nanostring who helped with this study, especially Robin Fropp, Lian Liang, and Kit Froman. Thank you.